First question from 2020. B. Okay. We are talking about photosynthesis. I hope you all have remembered what we are talking about, right? Option D. Okay. Anyone else? A. Okay. A. Okay. So we are talking about photosynthesis. So that means the normally if we are talking about photosynthesis, what happens? The plant will create its food and the oxygen emission will be happening. That is the first process. Two processes, light reaction and then dark reaction. Light reaction for the generation of the energy required for the dark reaction to happen. This is how we will solve the thing. So what happened in the light reaction? What is the significant things? Firstly, ATP, NADPH will be generated, oxygen will be released, water splitting will be happening, which is we will get from the stroma. That dark reaction that they will be recirculated, right? So from the water splitting, oxygen generation will be happening. So these are the key features, and the ATP and NADPH will be produced from this light reaction, will be ultimately used in the stroma, the enzymes present in the stroma region for the production of the carbohydrates as an energy source. This is a basic thing that will be happening. So that means the chemical intermediates that will be formed during this process will be ATP and NADPH. Right? These are the chemical intermediates which will be formed during the light reaction as a product. And even the one more option they can add, the oxygen will be also produced during the light reaction by the splitting of the water. So, but here in the given option, the O2 generation will be happening ATP and NADPH, not CO2. Next. Option C. D. A. B. So we have all options. Good. So reabsorption of which molecules in the kidney occurs in via passive transport. So two transport system that we are looking for. One is the active transport system and another is a passive transport system. So blood is flowing through the nitrogenous waste, means it has both nitrogenous waste and good material that can be remaining from the digestion process and still will be there. So two things will be happening. One is the active transportation, which is happening in the earlier part. That is the, if you remember the structure of the uh, tubular system, that will have the Bowman's capsule will be there. Then there will be the PCT, that is proximal convolutory tubule, distal convolutory tubule, and the convolutory duct. Convolutory tubule or convolutory duct that we called. So active transport is involved in the absorption of the remaining nutrients, amino acids, then the ion absorption will be there. So that things all, I mean, especially glucose and amino acid reabsorption will be happening in the PCT region, that is proximal convolutory tubule. In that part, and even in the glomerulus region or the Bowman's capsule, glucose reabsorption will be happening. But this is all the part of the active transportation. Then in the DCT, the ions which are essential part will be get reabsorbed. So many carbonyl ions, potassium ions, sodium ions will get reabsorbed in DCT. During the passive transportation, it will be help for reabsorption of the all the nitrogenous waste, which will be firstly during passive get reabsorbed in the convolutory duct and then given out in the as a part of the excreta in the last part of the process. So it is a nitrogenous waste. So among these options, the part of the active transport system reabsorption will be happening for the urea, which is a kind of a nitrogenous waste, which will be eventually given out in the from the kidneys to the excretion to happen at the later part. So passive transportation urea. Option C is the correct one. While the all other options are belong from the active transportation reabsorption. Next. Option C. Option B. Okay. Sorry, D that you are saying. Sorry. Option option D. Okay. So option D is the correct one because if we are saying G2 phase, so G1 is G2. So G1, the earliest start, there is a no division of the chromosomes will be happening. So number will be remain same. So uh, that way we can see the thing. However, in the G2 will be happening after S. So in the S phase, the doubling will be happening for the total DNA content. So the chromatid numbers will be doubled. So that will be ultimately the 92. So G2 phase, the 92 number of chromosomes will be there. Then G1 phase, the chromatid number will be remain diploid in nature. So that is 46 number of chromatids will be there. Spermatogonia is a pre-replication. So before starting the replication process, so spermatogonia is the cells which are present in the testes. We are diploid in nature, which will be after meiosis happen. It will be formed to the ultimately to the haploid sperms. So earlierly spermatogonia is diploid in nature. That means it again have the 46 number of the chromosomes. So B and C both have 46 and ovum is the haploid female gamete which have the half the number from the diploid cell which will be the 23. So D option 3. So option D is the correct one. Next. C. Okay. Yes, option C is the correct one because if we are talking about predator population will deplete, eventually the herbivores will be increased because of that, and that will be have the nutrition source from the trees, so that it will be the exploitation of the additional trees. So whole system will be get affected because of this. So that's why the ecosystem will be breakdown eventually if the process keep on continuing. So option C is the correct one. Next. Option. Okay. Yes, option D is the correct one because logical growth have the sigmoidal kind of a curve where first will be the lag phase, then the exponential increase will be there, and then as per resources is limited, it will be have the stationary phase. Exponential logical growth have this kind of a S kind of a pattern, and the exponential growth have the J kind of a pattern because there is unlimited resources is available. That is the basic assumption for the exponential growth. So option D is the correct. Next. D. Option A. Okay, so there are, when it comes to the animal kingdom, there are different kinds of a phyla which have the different kinds of a body symmetry. So three basic classes, one is asymmetrical, then there will be the radial symmetrical or the bilateral symmetrical. So I hope you all are aware with the, yes, okay. So if this is a case, so here is a means chart that you can refer to. So porifera usually this belongs to the class of the asymmetrical kind of a body symmetry they have. Then there will be the radical symmetry which is present in case of Coelenterata or Ctenophora. And then the bilateral symmetry is present in the all over the other classes from the chorddates to coelomates, acoelomates and all the things. So we are talking about the bilateral symmetry and the vertebrate system, which is the belong to cyclostomata, because cyclostomata is a class of the jawless fish. So it is a vertebrate which have the jawless fish. So fish have the bilateral symmetry. So they do not have any they have the bilateral symmetry, not the radial symmetry. So for this kind and the Nidaria and Stenophora, they have the they have already radical kind of a symmetry. Echinodermata is a special case that you can remember. Echinodermata adults have they have the radial kind of a symmetry, but their babies, when they are in the larvae stage, they have the bilateral symmetry. So this is a special case that exceptional case that you can remember. As per concern to this particular question, the cyclostomata will be correct answer. But echinodermata babies have the bilateral symmetry, babies or the larvae you can say more precisely. And once they turn into the adults, they have the radial kind of a symmetry. Next. B, C. We have actually just discussed this before in the other question in 2019. We are talking about muscle contraction, right? So for muscle contraction, calcium ions are very much essential. They depleted from the sarcoplasmic reticulum. The similar like the endoplasmic reticulum, the endoplasmic reticulum of the muscular cells have the it's rich with the calcium and they are moreover referred as a sarcoplasmic reticulum. So calcium is the essential ion or the crucial ion for the whole process of the muscular contraction. So option B is the correct one. Next. Option C. Yes, C is the correct one. Formation of mycorrhiza, this is a special symbiotic association between fungus and leguminous plants where the mycorrhiza or fungal hyphae are helping for the uptake or increasing uptake of the water, and they are helping for to get the minerals which are not usually water soluble to the available and make it available to the leguminous plants. Next. C. Option B. Option C. So here we are talking about a process of give rise to new organism from unfertilized ova. So that means the fertilization process should be absent. That is the very much key requirement that we are talking about. Xenogamy, autogamy are kind of examples of the pollination process that will be happening. That is the xenogamy happen between the two different plants. When the cross pollination will be happening, then it's called xenogamy. Autogamy, the fertilization process happening in the same flower from the same plant. That is the autogamy kind of. Parthenogenesis is a formation of the process of the formation having the new organisms or the new thing which we are in is the parthenogenesis process from the unfertilized ova. That is the new organism will be getting rise to the thing will be the parthenogenesis. Parthenocarpic fruit that we have developed. The parthenocarpic fruit have no it's a process of the parthenogenesis is involved for development of parthenocarpic fruit. However, as the fruit is the seedless, then we have discussed that they are not able to give rise to any other progeny. So the linkage cannot be the case. However, the parthenogenesis, the process by this process, the unfertilized ova can be developed as a new organism itself. In both plant and animals, this kind of a day. Some protozoals have this characteristic of parthenogenesis. Apomixis, the characteristic that we have just discussed about the apomixis, how the seeds will be formed without fertilization. This is here also the not fertilization process will be taking place. Syngamy is a pure fertilization process where two different gametes are meeting together. So that means the unfertilization, no, it's a fertilization process that will be happening. So parthenogenesis. Yes, option B is the correct one. Apomixis and parthenogenesis have helped to give rise to new organism, both in case of the plants and animals. This is happening. So I have provided some explanation for this, so you can get through it very clearly. Means how the syngamy is the process of formation of the zygote. Apomixis is without union of the gametes, the structure will be the form. That is the apomixis. Parthenogenesis, the this will be formed without fertilization again. Autogamy is the process where the pollination of the process, where self-fertilization happen, then we called it as a autogamy. Similarly, the for the biological means zoological perspective, it will be little bit different. And xenogamy is an example of the cross-pollination. Next, option B, option A, okay, B, yes, cry protein. Yes, yes. Now you can say sorry, but at the time of when you circle the thing in examination, please do remember and read the question and answers very carefully. That is very much important. So, Kaya protein is important for which is produced by the Bacillus thuringiensis, which we will be using as a in case of the BT cotton to develop. So, technology for which are resistant variety for the pests. Next. Option B. Option B is the correct one because amino acetylation happens in the cytoplasm once the process of the protein synthesis will be starting at that time, because amino acetylation involved for the activation of the tRNA to get charged with the particular kind of a amino acid get attached to it. So that is not part of the mRNA, functional mRNA formation. Splicing of the introns will be happening, which is the case of the functional mRNA to form. Polyadenyl tape will be added at the 3' end. That is also part of functional mRNA formation, and 5' end capping will be also happen with unusual guanosine nucleotide. So that is the D. So B is the correct answer. Next. A. Option A. So root modifications that we have seen various different kinds of a root will be additionally generated because of many reasons. Maize and sugarcane are the classic examples of the root modification with stilt roots, where the roots usually we have underground thing. These stilt roots, if you have ever seen the maize or the sugarcane plant, they have the from the lower nodes of their trunk, they have the some additional roots that we can see very easily at its base. So they give the additional support to the plant. That is the very characteristic of the maize and sugarcane kind of a sugarcane. These two plants only have this kind of a root, that is a stilt root. Prop roots is the another support system which one of the classic example is the banyan tree. So banyan tree, they have the roots from their branches which will be supporting the whole plant again, and they will be just from the branches they will arise and then again get attached to, once they get in contact with the soil, they will have they grow underground again and further development and a more support to the bigger plant. So that is the example of the prop root system. Fibrous and lateral root system are not, definitely not the case of the root modification. These are the types of the root modifications that we have the normal class belong to there, but stilt root and prop root, these are the two classic examples. Please do remember that. Next. A. Yes, A is the correct answer. Glycerides are made up of two things, means glycerol and fatty acid. That makes the glycerol. Glycerides. So diglycerides have one molecule of glycerol and two molecules of fatty acids. That makes the diglyceride. If we say triglyceride, one molecule of glycerol and three molecules of fatty acid. Like so, the nomenclature will be there. So in the amongst the option A will be the correct one. Next. Anyone? Option D. Okay. You all know Hardy-Weinberg equilibrium equation. Okay. So Hardy-Weinberg equilibrium is mainly stating about the allelic frequency or which will be happening in a particular population at given time will be remaining constant through from generation to generation. This is what is. So that allelic frequency, though it is changing the format, that can be because of the various factors like genetic drift, mutations, that can happen, which can affect the various equilibrium between the Hardy-Weinberg equilibrium. But the main functionality that we can talk about is the main, it is a, when we want to put equationally two factors. So the first equation that the Hardy-Weinberg is the P plus Q bracket square is equal to 1. This is the main thing that they have stated. That is P square plus 2PQ plus Q square is equal to 1. And the one that you are saying, the percentage of the homozygous recessive individuals or the frequency of the dominant allele in the population and frequency of the recessive allele in the population is equal to 1. So both things we should consider when we want to solve this kind of an example. So here the frequencies that we are representing with this, then P square is the percentage of the homozygous dominant individuals, Q square will be the percentage of homozygous recessive individuals, and 2PQ will be representing percentage of heterozygous individuals, which we need to find in this question. So we have the number of the colorless, that is the recessive one. That is the q square value we have, which is a 490. So firstly, we want to find the fraction of the q square. So amongst the 1000, 490 will be having this particular colorless thing, that is recessive one. So its frequency is a 0.49. So if we want to calculate the value of the q allele, that will be the square root of the 0.49, which is the zero point 0.7. So then we will consider the frequency of the both alleles. Then we can have the value of the p, which will be the zero point 0.3. Then we get the frequency. Now we have got the frequency of the dominant allele and frequency of the recessive allele. After that, heterozygous flower will have the 2pq, that is its fraction in the given heterozygous environment. So by considering these value, we will get the 0.42. So now we want, this is the frequency that we have got the heterozygous red flower population. So we have total hundreds. Among this is the frequency of the heterozygous one. So we get 420. So 420 is the number of the heterozygous population with the means capital R, small r, that heterozygous kind of thing. The genotype can be defined based on that. So this is the total number of the heterozygous red flower in the given population. So 420, that is option C is the correct one. Last question. Sorry, D. Okay, D. D. Okay. Okay. Anyone else? So what happens when virus infected a particular cell? If the virus infected a particular kind of a cell, it produces the proteinaceous chemical substance or proteinaceous substance, which we call the interferon. So it is produced by the virus infected cell. What's its main function? Because cells are lined up on the near to each other. So once the interferon gets secreted from the virus infected cell, it helps to get immunity to other cells. So it's protect the other uninfected cells from causing the virus infection, and eventually the virus infected cells get died, but the remaining cells will be get protected. So main function of the interferon is to help the or protect the uninfected cells. That is the one thing. Here, the lymphocytes won't play any role. So B lymphocyte, T lymphocyte do not play there. This is a special thing which is secreted only after virus infection. So interferons is a class of protein secreted by virus infected cells and are involved in protecting uninfected cells. So option B will be the correct one. Lymphocytes are not involved in the production of the interferons. So please do remember that.